So it is my pleasure to introduce the next chair of this last session, Ramiro Pastorinho from University of Évora and the Comprehensive Health Research Center. He is also a partner in the IonCytDevice project. And we are going to address now in this final session the treatment for cancer, and hopefully with the research conducted by the speakers, we will have really nice insights on the future perspectives that science is bringing us on the new treatments for this problematic disease. So, Ramiro, if you want to start the session, please.
Thank you, Anna. Good afternoon, all. Here we come to the last session. I hope you're not tired. There are still many good things to come. So basically, I'm going to start and introduce Iola. I would just call your attention upon the fact that if you have any questions after Iola's talk, you can raise your hand and I'll tell you to put your camera on and your audio on, and you can formulate your question. After that, please turn them off again. I just remind you that the workshop is being recorded and it will be available in the YouTube channel of PATH. So the first speaker is Iola Duarte. She's a principal researcher at CICECO. She is the recipient of the prize L'Oréal PT for Women in Science and the Portuguese League Against Cancer grant. She is interested in researching immune metabolic deregulations in chronic inflammatory diseases, biological responses to nanomaterials, and tumor metabolism, anti-cancer drugs, and nanomedicines. Her talk will focus on the metabolic profiling of cancer cells and the modulation of the metabolic phenotype axis in tumor-associated macrophages. It is entitled Metabolic Profiling and Modulation in Cancer Treatment. Iola, the floor is yours.
Thank you. So I'll share my screen. Can you see the presentation already?
Not yet. Yes, no,
okay.
It's on.
It's displaying, okay. Well, thank you so much for the introduction. Also, I would like to thank the organizers, and in particular, Marcia Neves and Mara Freire for inviting me to be here to participate in this workshop, which I find a very interesting initiative, especially because of its multidisciplinary nature, because so many different aspects of this complex disease are being touched and are being discussed here. And so I'm not sure if I will deliver what Anna Catarina said about new ways to treat cancer, but at least I hope to give a contribute by talking about deregulated tumor metabolism and how this property of cancer can be harnessed for additional treatment strategies. So I'm a chemist with a special interest in biochemistry, basically, and human metabolism is one of my interests. And this is because I find it fascinating how tumors can really adapt and change their biology, including their energetics, their cellular energetics. We already heard this morning about the tumor enabling characteristics and hallmarks of cancer. So this is just a brief overview of the main hallmarks of cancer that I'm showing you here in this figure. So cancer cells, basically, they are able to be self-sufficient in the production of growth signals, and also they switch off the growth suppressors. They are able to become immortal. They avoid the destruction by immune cells. They also are able to invade the surrounding tissue and to spread to distant body parts, so they are able to metastasize. They are able to promote the formation of new blood vessels to provide them with nutrients and oxygen. They also bypass an important mechanism that we have in normal cells, which is programmed cell death, the apoptosis, which happens when cells become damaged in some way. And cancer cells are very resistant to apoptosis, and they are also different in their metabolic program. So they change their metabolic circuits in order to provide survival advantages, in order to provide energy and sustain their biosynthetic needs and survive in a harsh environment. Sorry. And the first insights about cancer metabolism actually came more than 80 years ago by Warburg, and it became known as the Warburg effect. And just to give you some context, so while normal cells process glucose into pyruvate in the cytosol in a process called glycolysis, and then they further oxidize pyruvate into carbon dioxide in the mitochondria to produce ATP, to produce energy through oxidative phosphorylation. The tumor cells have a preference for glycolysis and a downregulated oxidative metabolism, even in the absence of oxygen. And so this became known as the Warburg effect. Of course, at the time, Warburg was far from knowing that this was only the tip of the iceberg. And we now know that cancer cells change their metabolism in many different ways and affecting different metabolic pathways involving glycolysis, pentose phosphorus pathway, involving TCA cycle, oxidative phosphorylation, the metabolism of lipids, amino acids, and so on. And this is still something that is unveiling as research goes on. We also know that there are different reasons for this altered tumor metabolism: genetic and epigenetic changes in oncogenes, tumor suppressors and enzymes, and also changes in the tumor microenvironment and interactions with the stromal cells in that environment. And this is not just a mere consequence of these factors. Actually, this tumor metabolism, we know now that it is very active in promoting tumor survival, proliferation, invasion and metastases, the suppression of immune cells, effector functions, and even drug resistance. And so the natural corollary from this is that it is important to know which metabolic disarrangements we have in tumors that are related to all these consequences. And I'm sure that you know that we already have important developments related to this altered tumor metabolism. For instance, in FDG PET imaging, which is an imaging technique based on the fact that tumor cells consume glucose, in this case an analog of glucose, at a much higher rate than normal cells. And in this way, we can localize malignant lesions or even follow their progression through treatment. And another important development that is coming up from the knowledge of altered tumor metabolism is the development of several anti-cancer drugs targeting specific metabolic pathways and enzymes. And most of these, this is just a short list of some examples. Most of them are in phase 1 and 2 clinical trials, although, for instance, this one here, targeting isocitrate dehydrogenase 2, is already approved by FDA. It was in 2017, if I'm not wrong, and to treat acute myeloid leukemia. Also, phytochemicals, plant-derived compounds, are emerging as important metabolic modulators, which can have anti-cancer properties. And this is also one of the areas where I'm interested, and I've worked recently with a PhD student. I will show you some of those results in a little bit. So, of course, this search for metabolic dysregulations and targets can be a very difficult task, and we need really tools that can approach us from the ideal world of finding these dysregulations and targets. And so this is where I think that the omics technologies come in very nicely. They are part of the so-called systems biology approach, and we have different levels of biological organization to which we can look at. So from the genome to the mRNA transcripts, to the proteins, and to the metabolites. And my focus is on metabolomics. It's the main omics technology that I've been using. And so I will just briefly tell you something about it before I will share some examples of our work. So we can define metabolomics as the systematic measurement of metabolites, and metabolites are these small molecules like amino acids, fatty acids, carbohydrates, and so on. They have a molecular weight under 2 kilodalton, and their cellular concentrations can vary from picomolar to millimolar. And so they are closely related to proteins and genes, and the big difference, I guess, it's this one. So if your genes can tell you what can happen, the metabolites give you a more exact picture of what has happened and is happening. So it's a very close level of information, or it's a level of information very close to the final phenotype and function of cells. This is an overview of the metabolomic strategy that is usually employed. So we start by analyzing biological samples, which can be biofluids, tissues, cells, cells growth medium, by analytical platforms that allow us high throughput screening of a high number of metabolites at once. And these platforms are NMR spectroscopy or mass spectrometry-based methods, usually. Of course, there are others, but these are the main ones. Then we end up with spectral profiles, which are very complex, and so we need multivariate statistics and other data treatment tools to identify the metabolites present, but also to assess the differences between sample groups and to highlight the effects that we are looking for. Then there is the very challenging task of biological interpretation. This is especially challenging because metabolites are usually substrates and products which participate in several metabolic pathways, and so it is difficult to infer immediately just by an increase or a decrease of a metabolite what that may mean in terms of the biological network where metabolites are involved. So usually we need to generate hypotheses and then do some experimental validation with complementary tools or even with metabolic flux analysis. Okay, so this was an introduction, and now I will show you some results. I'll try to keep on time. And the first study that I'd like to show you is a study that we've done in collaboration with Iola Duarte from CEBAL. Angela Guerra was our PhD student. She finished her thesis last year. And her work was about the modulation, metabolic modulation of breast tumor and epithelial cells by a lipophilic extract from eucalyptus bark. And just before I go into these results, I'd like to give some context. So this work really started almost 10 years ago, and it did not involve me at the time. It was, but I think it's important to mention that we have a group, Carmen Freire and Armando Silvestre, for instance, who are at the department, who is very strong in this kind of work where they isolate bioactive compounds from natural sources. And in this case, this was they started this work by isolating or extracting bark from eucalyptus and characterizing its composition, identifying the main bioactive compounds where the three terpenoids emerged as the main constituents. And so they went on showing that these extracts from bark had actually very interesting bioactivity properties, and including anti-tumoral activity against different cancer cells. And so they started working with triple negative breast cancer cells. This is a very aggressive type of cancer, which is very resistant to different kinds of treatments. And so there is a great need for new strategies to fight this specific type of breast cancer. And so from this basis, we did some work to know what was the impact of the eucalyptus extract on both the triple negative breast cancer cells and epithelial breast cells, non-tumor. So we did some in vitro studies where we have collected the medium and we had extracted the cells to obtain two phases, the aqueous and organic, and then we did the analysis by protein NMR. So here I'm just showing you the kind of record that we obtain when we look at an aqueous cell extract. So these signals correspond to the different metabolites present. We can detect several organic acids, amino acids, nucleotides, sugars, and several other metabolites. In the organic extract, we have lots of signals from lipids. They can be membrane lipids, such as phospholipids, or they can also be neutral lipids, which are typically present in the cells, cytosols, as lipid droplets. And then if we look at the culture medium across time, we can see what the cells have consumed and what they have excreted, and how this differs between, in this case, between normal and cancer cells. And so overall, we could easily identify a number of metabolic features typical of the cancer cells: upregulated glycolysis, low mitochondrial respiration, low ATP levels, and also low NAD plus to NADH ratio, high phosphocholine, and lipid droplet content. And so the next question was, okay, so how is this modulated by the eucalyptus bark extract? And we acquired, after acquiring the spectra, just to give you an idea of the strategy, we then can run well, easily the multivariate analysis, where, and you see here these plots, these scores plots, where each point represents one sample. So we have a clear separation between control and between control and the treated samples. And we can find the signature, the metabolic signature that explains this separation, and the same for normal cells. And from here, we can already see that the effects, some of the effects are common, while others are different. So next, we look into more detail about these variations. And here you have a summary heat map where we have a color scale reflecting decreases and increases in relation to controls. both for cancer cells and for epithelial cells. And so we can see here, for instance, decreases in TCA cycle intermediates, increases in a number of amino acids, and other variations. I will not go into detail. I don't want to bother you with this. So what I want to say, however, is to highlight some changes that were unveiled in this study. And one of them was actually a strong decrease in the phosphocholine to choline ratio. And why is this relevant? Well, the accumulation of phosphocholine in breast cancer tissues is considered to be a biomarker, actually, of breast cancer aggressiveness. And it is related to the increased activity of this enzyme, choline kinase. And so we found that the extract could really downregulate the expression of choline kinase A, and so this can probably explain this great decrease. And another significant change was this modulation of the NAD plus to NADH ratio. So this ratio typically reflects the balance between oxidative and glycolytic metabolism. So as I try to simply illustrate here, while glycolysis and the TCA cycle consume NAD+, this NAD+ can be regenerated through oxidative phosphorylation in the mitochondria during the production of ATP. And so cancer cells, which are mostly glycolytic, are characterized by a low NAD+ to NADH ratio, whereas the other normal cells with a functional oxidative phosphorylation have a high ratio of this cofactor. And what we see also from the literature is a negative correlation between this ratio and the aggressive malignant phenotype of triple negative breast cancer cells. And also there was this study that showed that enhancing this ratio could actually lead to inhibition of tumor growth, both in vitro and in vivo. And so this is a positive outcome from the treatment with the initins extract, as we see that we go from a low NAD plus NADH ratio, so a low reductive power, to an increased one close to control non-cancer cells. Okay, so this is an overview of all the changes that we've seen, considering the consumption and expression patterns, and also the intracellular profiles. And basically, to give you a sum up, this eucalyptus extract was able to shift the metabolism of cancer cells to mitochondrial respiration. So there was kind of a restoration of the normal mitochondrial function, fueled by fatty acids and amino acids. We also saw activation of the exosamine biosynthetic pathway and protein degradation, which are actually consistent with the activation of the unfolding protein response in its pro-survival form. So actually, we think that this reflects, this requires confirmation, but we think that this reflects the induction of ER stress, which was able to boost mitochondrial metabolism of cancer cells. And in that way, it could be viewed as hitting a vulnerability of cancer cells because it kind of reversed a major malignant phenotype, which is the Warburg effect. So here in green now I am showing you the changes induced by the same eucalyptus extract but on epithelial cells. And here we also see some metabolic features of ER stress, but we see upregulation of glycolysis, so a different profile from the cancer cells. So I've told you this already. We saw a Warburg reversing effect on cancer cells, and this opens potential to treat this kind of cancer by hitting the vulnerability of cancer cells in metabolic terms. But there was a major problem with this study, which was the highest toxicity of the extract, the eucalyptus extract, in epithelial cells compared to normal cells. And of course, this is a limitation due to the secondary effects that can be induced if we consider in vivo work with this extract. And so we ask whether pure tritopanic acids could induce similar effects with lower toxicity. So we have tested, at this stage, we have tested betulinic acid and ursolic acid, which are two major tritopanic acids of the eucalyptus extract. We did see that the toxicity, the cytotoxicity in this case towards breast cells was actually similar for for cancer and for non-cancer cells. So this is good news compared to the whole plant extract. And then we went on to do the same kind of approach to see what was the metabolic impact of these two compounds on both types of cells. And summarizing the outcome, we could see that betulinic acid actually had a much stronger impact on the cell's metabolism, while the effects of ursolic acid were really mild. Betulinic acid did have some common effects compared to the initins extract, but also distinct effects, and no change in the oxidative metabolism. So we don't think that betulinic acid alone is able to achieve that effect, and we probably need a combination of compounds to recapitulate what we saw with the extract. So this is work to continue. Okay, so I'll switch now to the second example. I hope I have how many? 5 minutes? Okay, thank you. So in this other study, which is a very recent work from a PhD student, Ana Dias, co-supervised with Catarina Almeida and Luís Elguero from IBMED, also from the University of Aveiro, we are looking into the modulation of tumor-associated macrophages. And so just to give you some context, these macrophages are immune cells that are present in the tumor microenvironment, and they have been associated with tumor-promoting features such as immune suppression, angiogenesis, and tumor growth. And so one of the ways that people are thinking to approach cancer treatment is through macrophage repolarization. That means converting these macrophages into macrophages with the ability to fight, or with better abilities to fight the tumor cells through immunostimulation, antigen presentation, and T-cell activation. And there are many ways to promote this. We are looking into the promotion of these repolarizations through metabolic reprogramming. And this is because in the last years, there is emerging evidence that metabolism really is a central axis of macrophage phenotypic and functional regulation. So there are many recent papers starting in 2013, I think, showing that different immune cells and different activation states of immune cells like macrophages need a different metabolic program, and this is strictly related to their function, to macrophage functions. And so we are addressing this issue in this project of Anna, and we are starting by the in vitro generation of a TAM model, metabolic profiling to identify metabolic targets, and then we will proceed to modulate these targets and see what impact we have on cancer hallmarks like invasion and angiogenesis. As I said, the project is at the beginning, so we are focusing on the establishment of a TAM-like model. And for that purpose, what we are doing is to incubate macrophages derived from human monocytes with growth medium conditioned by breast cancer cells. And what we have seen so far is that we did have some phenotypic features which are characteristic of TAM-like cells, like increased expression of the interleukin 10 and NOS2, and in the metabolic component, we see, well, so far we have looked, or here I have the results only of the exometabolome. This means metabolites consumed and excreted by cells. We did not look at the intracellular profile yet. What we can say at this moment is that these macrophages, when they are conditioned by tumor cells medium, they indeed change their phenotypic and metabolic profile. In particular, they reduce the catabolism of branched-chain amino acids and increase TCA cycle or the citrate export. And so these are some things to look further in the future to assess if the modulation of these regulations can have some effect on the function of macrophages in the tumor microenvironment. So, as final remarks, I hope I'm not over time too much. Just a simple take-home message. So, tumor metabolism and the metabolic crosstalk within the tumor microenvironment contribute actively to tumor progression and also its response to treatment. Metabolomics of tumor and tumor-associated stromal cells, like immune cells, can be useful to reveal metabolic vulnerabilities and also some pharmacological targets that can be tested in drug screening platforms. I believe that bioactive compounds directly targeting dysregulated metabolism of tumor cells, or probably even more efficient, modulating the tumor microenvironment, are expected to aid anti-cancer treatment. So thank you for your attention. I also would like to thank my students, collaborators, especially in those projects that I've told you about, the UA and MR Metabolomics Lab, my colleagues, and also, of course, the funding entities. Thank you again for your attention, and I'll be happy to answer your questions and discuss a little bit.
Thank you, Iola, for your excellent presentation. I would now ask if anyone has questions. Please raise your hand. Okay. João T, Barata, I would like you to please turn your camera on and microphone.
Yeah. Hi.
Hi.
Hi, Ramiro. First of all, nice to
Hello.
see you. And a very nice talk. I have a few questions regarding both the first part and the second part of the talk. Maybe I start with the second. I was a bit surprised to see that these in vitro educated TAMs, so produce IL-10. IL-10 is normally associated with an anti-inflammatory profile, not a pro-inflammatory profile that normally would be, you would expect, for, in a protumorigenic context. So, do you have an idea of why this happens in your in vitro culture system? This is the part, and the second part of the question relates to whether you know whether the production of IL-10 is linked to rewired metabolism or not, or if the two things sort of go hand in hand in parallel, but are not. Do you have any data?
Okay. Well, thank you for your question. Actually, I think that although there is not a straight line to characterize exactly which phenotype do the tumor-associated macrophages have, I think that there is some consensus now that they are mostly anti-inflammatory, so M2-like macrophages. And so one of the features that we expect is that they increase the expression and production of IL-10. Yes, so that result was actually in line with what we expected to have. Although, as I said, it's very difficult to characterize in vivo, so we are just based on the in vitro results. As far as I know, the link to metabolism is not completely clear. So, for instance, in the case of pro-inflammatory macrophages, there is some evidence showing already a link between, for instance, the production of succinate and stabilization of the hypoxia-inducible factor and the production of IL-1 beta. So this link has been unveiled in some circumstances, in some activation setups, but I'm not sure, but I'm not aware of the link yet between the production of IL-10 and specific metabolic pathways. But it will be interesting to look at that.
Okay, I have another couple of questions, if I may. I don't know.
Yeah, sure. Go ahead.
So regarding the first part, which and the eucalyptus extracts. One of the things, of course, then you sort of, because of the toxicity effects on normal breast epithelial cells, you sort of shifted away from the extracts. But given that the tritopanic acid results are not that as promising, I would say. I think it's still interesting to sort of dwell on the role of the extracts as a whole. And so my question relates to whether you think it may be a problem, in addition to the toxicity issue, the fact that you've seen that the extracts apparently in normal epithelial cells shift the metabolism towards glycolysis. So in a way, they are recapitulating, in a naive perspective at least, what I would expect from the Warburg effect, right? So you would kind of, do you consider that could be sort of a danger of the use of these extracts or not? And yeah, I have another question, but maybe.
Okay, maybe I can comment on that first. Yes, I think that's part of the problem, and probably part of the toxicity also, because if the extract is affecting the mitochondria and damaging the mitochondrial activity, then maybe glycolysis, it's the increase in glycolysis is a compensatory mechanism, but still it may not be very healthy for cells. So yes, I think that could be a problem, and I think that we cannot exclude. Also, I think that we should not put it away immediately, and maybe we have to think of this kind of plant extracts and compounds integrated in other approaches like the more targeted delivery to the cancer cells, because I think it's really difficult that we will find compounds that or plant or other extracts that will specifically hit the tumor cells without having damaging effects in normal cells. But yes, well, the theory behind it is good. That is that, well, okay, if the cells have different metabolic programs, then maybe we can selectively affect cancer cells without damaging the normal ones. But so far in the work that I've done, unfortunately, I didn't see that happening in a very in a straightforward way. There is always shortcome.
But yeah, that sort of leads me to my last question. I don't want to monopolize the discussion, but which relates to the opposite observation on cancer cells, that you sort of reshifted from the glycolysis to mitochondrial respiration. And so this is a very naive question from a guy that doesn't work on metabolism at all. But would that open the possibility of combining, for instance, low doses? I know that high doses would be absolutely toxic, but low doses of respiration blockers, for instance, together with eucalyptus extracts. Would that be something that you would consider as really useful?
Yes.
In a way, you're rewiring the survival of the cells from glycolysis.
Yes, I think that is the way to go. Actually, it's not to kill tumor cells directly, but with this kind of modulating strategy, I think we can hit a vulnerability. So glycolytic cells they have a lot of survival advantages, and one of their vulnerabilities is not to have or one of their ways of resisting common anti-cancer agents is through that altered metabolism. So yes, if restoring, I understand that restoring the mitochondrial function could be a way to make the cells more susceptible to other treatments that hit mitochondrial respiration. Yes, and there are some works already showing that it is a good way to go. Thank you. Hope I've answered.
Okay, we still have time for a very, very, very brief, rapid question. Anyone? So I'll ask something. When you presented the results from the extracts, you didn't spoke about any triterpenic acid specifically, and you presented a lot of results. Only towards the end, you spoke about specific triterpenic acids. The first results you presented were regarding the extracts as a whole, or were they like specific triterpenic acids being tested?
No, they were a lipophilic extract from the outer bark of eucalyptus.
The whole extract, right?
The crude extract. Yes.
Okay. Another thing, and bear with me because I'm a poor ecotoxicologist, I don't know anything about this, but the thing is that you told us that you had a lot of ER effects, right? But
it looks like.
Yeah, but you spoke a lot about like the metabolism of sugars, and ER doesn't have too much to do with that, does it?
Well, I think that evidence is emerging that it does, actually.
Oh, really?
And that, yeah, and that
because classically it's lipids and proteins, right?
Yeah, but also I think that energy metabolism has a role here, especially when the cells are adapting to restore proteostasis, and they need also to shift their energy ways of the ways of producing energy. I found there is a recent paper on how with ER stress cells can form, well, they form super complexes. In the ETC chain, the electron transport chain, they form super complexes. They assemble, and also there is an interesting study showing how mitochondria get in close proximity with the endoplasmic reticulum during ER stress. And so this all suggests that it's also carbohydrate and energy metabolism has a role here, and not only protein and lipid metabolism.
Okay, excellent. Thank you very much. Iola, thank you very much for coming to us and presenting this very interesting talk. Now I'm going to give the word to João. Sorry to be so informal, but that's been the theme of the thing up till now, so I'll keep it up. João, could you please connect your video and audio again?
Sure.
Okay. So it is my pleasure now to move to the next presenter. It's João Barata. He's an associate professor at Faculty of Medicine of Lisbon University and principal investigator at Institute of Medicina Molecular. He was a recipient of an ERC consolidated grant and proof of concept. His lab, he has a lab, he's a fancy guy, focuses on studying the signal transduction pathways that are deregulated in cancer by cell autonomous lesions and microenvironmental cues, with the ultimate goal of identifying new targets for therapeutic intervention. During his talk, he will provide an overview of his lab's most significant achievements on acute lymphoblastic leukemia. The talk is entitled Metabolic Profiling and Modulation in Cancer Treatment, and you have, no, it's not, okay, Len, correct me, please. So go ahead, the virtual floor is yours. Thank you very much for coming.
Thank you so much, Ramiro. So it's really a big pleasure to be introduced by a good old friend of mine and someone that I respect scientifically a lot, although in a different, working in a different area. So I'm very pleased by that. I'm also pleased with the invitation. So I thank very much Mara and Kati for the invitation and Souza. I've been there already, fortunately, physically some time ago, and it was a real pleasure. So it's quite nice to be back. On the other hand, I'm afraid I will repeat a couple of things that I've presented before, not totally because of the sake of time and also not to bother you too much. So I'll try to show some other stuff that we do in the lab that I didn't show the last time, for those that were present then. Maybe I should show Ramiro, that I also have comic books, a shelf with comic books, okay, somewhere there. I feel so it's a bit awkward to make a presentation, without seeing the faces of the audience. I'm going to try to share my screen now. So this is the actual title of the talk. All right. So I'll start, with a broad introduction about and call your attention to the fact. I think I'm okay. I see the time there. I don't want to go too much beyond time because I know I'm the last one. So I really don't want to be pain in the neck for everyone. But let me start by just calling your attention to the fact, and I always do this in my presentations, that cancer is it's really a heterogeneous disease, and we should look at cancer, in a broad perspective, and remember that besides cancer cells, which are heterogeneous in themselves, we have, as already referred in the previous talk, other types of cells that associate with the cancer, which are supposed to be normal, but that actually can contribute to cancer progression, such as cancer-associated or tumor-associated fibroblasts, pericytes and endothelial cells that constitute the vasculature, that sort of feeds on the tumors, immune infiltrates of different kinds of flavors. So because of this, it's obvious that the cancer cell is in a permanent dialogue with its microenvironment. And this dialogue is really essential to how a tumor evolves and whether a tumor may actually regress with time instead of developing into full-blown malignancy. Now, this crosstalk, of course, relies on the activation or inactivation of biochemical pathways that we call signal transduction in a large sense. And this cartoon is sort of very busy, but it boils down to a very simple idea, which is we have external signals that will activate receptors on the membrane, on the cell membrane. These receptors will then turn on biochemical cascades, which we call the actual signal transduction within the cell, that will then regulate a number of transcription factors or other proteins in the cell that will regulate stuff like apoptosis, migration, growth, adhesion, differentiation. So the very processes that are at the core of cellular homeostasis. And as a consequence, it's obvious that if these pathways are deregulated, we may have a problem, an health problem, including, of course, cancer. And so, as Ramiro has already pointed out, we're very interested in understanding how signal transduction pathways work in the context of cancer, precisely because they lie at the interface between what goes on in the cellular milieu or in the microenvironment and what goes on inside the cell. So if something, and I'll illustrate this further down the line in one part of the talk, if something goes wrong within the cell, there are signaling pathways that sort of detect that problem and try to cope with it. And in fact, the signal transduction pathways themselves, or different elements of the pathways, are so important that they are themselves subjective subjects of oncogenic hit. So they are quite often oncogenes or tumor suppressors. So the idea of the lab, and that of many other labs in the world, in fact, is to try and understand signaling, try to block the signal pathways that are really essential for cancer progression, and in doing so, trying to develop targeted therapies that can eliminate cancer. And so if we shift sort of from this slide to this one, which is more complex, we start to go closer to what we are doing in the lab, or what we have done throughout the years in the lab. And so I'll give you sort of an overview of some of the stuff that we've studied, starting with this pathway here, PI3 kinase Acute pathway, that can be activated by external stimuli such as IL-7, and I'll talk about this. IL-7 binds actually to the IL-7 receptor and activates different signaling pathways, including PI3 kinase pathway. But PI3 kinase Acute pathway can be activated also in a cell autonomous manner. So it can be aberrantly hyperactivated from within, let's say. And this is what I'll talk about in a few slides further down the line. Before that, I would just like to sort of introduce the disease that has been the main focus of our interest throughout the years, acute lymphoblastic leukemia. Actually, we focus more on TALL, which is one of the subtypes of ALL. And as you can see in the slide, overall the disease is constituted by these two different subtypes, either B-cell precursors or T-cell precursors that get arrested in their differentiation and have and undergo clonal proliferation. ALL as a whole is the most frequent type of cancer. It accounts for around a quarter of all pediatric cancers, and the vast majority of childhood leukemias. Now, TALL in particular, it's less common in children. It's something went wrong. I think my internet connection probably screwed up things, I guess.
No, we can still hear you and see you, so it's not internet connection. Maybe it's the computer.
That's
okay.
Great.
You're back again.
Yeah. Okay. Sorry about that. Because,
but you have that square again.
Yeah.
Okay. Excellent. Now it's good.
Okay. So as I was saying, TLL, the frequency of TLL cases it's lower than BLL, but these cases are higher risk, poor prognosis than BLL, and so there are two issues with that. First, there is still a significant number of relapses that occur. The good news is that with current treatment protocols, children can be free of disease after 5 years in up to 90% of the cases. So therapy, it's actually quite effective, but you still have 10, 15, 20 or more percent of kids that either do not respond to therapy or that end up relapsing. And when they do relapse, the prognosis is very poor. And in addition to this, the conventional therapy, particularly in high-risk cases, is very aggressive. And the consequence of that is that you have long-term severe toxicity. So it's commonly accepted that we need to develop new therapies that are both more effective in eliminating the cancer cells and less detrimental, that is, that affect less the normal cells. Okay. So with this in mind, I would like to now share a bit about our stories on PI3 kinase and AKT and how, because of that, we end up coming to know this guy here, CQ2. So PI3K AKT pathway, it's a pathway that is involved in cell proliferation, viability, migration, cell size increase, so increasing metabolism. It's not surprising that this pathway is very frequently hyperactivated in cancer. And it is hyperactivated by a number of different mechanisms that include gain-of-function mutations in PI3 kinase, in AKT, but also loss of function mutations in the negative regulator of the pathway, a phosphatase that it's called PTEN. So what PTEN does, it's precisely the opposite of PI3 kinase, which is a phospholipid kinase that basically phosphorylates PIP2 into PIP3. I won't go into the long name of this, but we can talk about that in the end if you want. PTEN dephosphorylates PIP3 into PIP2. And in doing so, it basically blocks the pathway. Now, PTEN is frequently inactivated in human cancer as a consequence of genetic lesions most of the time, sometimes epigenetic alterations that silence PTEN. And ultimately, what happens is that you have diminished, in most cases, diminished or absent mRNA control. So you have loss of expression of PTEN that associates with hyperactivation of PI3 kinase pathway. Now, what happened in TLL was that we identified this many years ago. This is an old story, but I still wanted to show you this because it's important for other stuff that we did afterwards, more therapeutic related. What we found out, if you look here at the levels of phosphorylation of AKT, which basically mean activated AKT, in TLL patients, as compared to normal thymocytes, which are the normal counterpart of these guys here. And if you look at the levels of phospho-AKT, you can immediately see that levels are much higher in the TLLs than in normal thymocytes. And this is exactly the same. So this is summarized here. The levels of phosphorylation of AKT, of activation of PI3K, AKT pathway, are quite high in TLLs. And we found that around 80 to 90% of the patients actually display hyperactivation of PI3 kinase pathway. Now, we also found, in a way, this is expected. Okay, these are cancer cells. As I told you, this pathway is very frequently hyperactivated, so no big surprise here. Also, not a big surprise when we realize that some patients hardly had any PTEN or no P10 at all, this conformed to what one would expect. But where things started to get really puzzling and interesting, as science should be, is when we realized that P10 levels in some of the patients were higher than the levels of P10 in normal thymocytes. And these patients, for instance, if you look at patient number 2 with high levels of P10, it's precisely one of the patients that has high levels of PI3 kinase AKT pathway activation as well. Okay, so we had in parallel two things that didn't make sense. This is just a summary of what I just told you. So a lot of patients had very high levels of expression of P10, which is the negative regulator of the pathway. It is a tumor suppressor. So P10 should be down in order for PI3 AKT pathway to be up. And that was not what was going on. They were both up. So to summarize a very long story in a slide, what we found out was that, in fact, some of the patients, 10 to 20% of the patients, do have genetic lesions in P10 that inactivate it and lead to loss of expression of P10. But the majority of the patients actually have this paradoxical event in which P10 is overexpressed. But overexpression of P10 actually leads to decreased P10 activity, and decreased P10 activity, of course, leads to high levels of activation of the pathway. So in a way, in a very paradoxical manner, high levels of P10 equal or are similar to loss of expression of P10. Why is this? Because I'm rushing. CK2, casein kinase 2, which is overexpressed in TLL cells, leads to the phosphorylation. This is what we showed in this paper in 2008. This leads to phosphorylation of P10 at its C-terminal tail. It makes P10 inactive, but at the same time, it stabilizes the protein. And because of that, because although we have a lot of P10, P10 is inactive, PI3K signaling is on, and so leukemia cells survive. If we use CK2 inhibitors or genetically deplete the cells of CK2, what we have is a reversal of this process. So we have loss of phosphorylation of P10. P10 becomes less stable, so we have less P10 in the cell overall, but it's more active. And this is sufficient to turn PI3K signaling off and leading to leukemia cells. So because of this, what we did next in other papers was to, in other studies actually that led to papers. Papers come from studies and a lot of effort, of course. What we did next was to try and push these findings closer to the clinic and go for in vivo preclinical models. What we did was to subcutaneously transplant TLL cells into immunodeficient mice so that they could accept this graft and not reject it, and then either leave the tumors untreated. And as you can see here, this is tumor volume, so it means that the tumor is growing, and it's growing exponentially, very rapidly. If we leave it untreated, if we treat with a PI3 kinase inhibitor that is clinical grade, you can see that there is a very significant delay in tumor progression. And this is another model in which the delay is not as striking, but there is also a delay. And we can prolong the survival of the mice that have the tumor graft. So in a way, this tells you that PI3 kinase inhibitors may be of interest clinically for treatment of TLL. Similarly, if we use a clinical grade pharmacological inhibitor of CK2, so the kinase that phosphorylates P10 and inactivates P10 and drives PI3K signaling hyperactivation, you can see here, this is the vehicle, this is the drug that inhibits CK2. The intensity of the signal is proportional to the intensity to the tumor burden. And you can immediately see that the mice that were not treated have more tumor than those that were treated. And you can see here in a quantifiable manner. And so, again, targeting of CK2 seems to indicate that there is indeed clinical potential for the use of CK2 inhibitors in TLA. Now, the problem we found, or something that we thought about, relates to the fact that CK2 is a very promiscuous kinase. It phosphorylates a lot of stuff in the cell. And so if we use a CK2 inhibitor, we may target the tumor cells, but we may also target normal cells. And although our studies with animal models actually didn't show much toxicity with the CK2 inhibitor, that was something that worried us. And so we thought, why not go and try to identify regulators of this link between CK2 and PTEN that can specifically abrogate the ability of CK2 to phosphorylate PTEN and not other stuff that CK2 does to the cell. So to conduct this sort of analysis, we did an shRNA screen in which we silenced all the kinases in the human genome, so what we call the kinome, and a few more genes. And our rationale was that probably CK2 is regulated by phosphorylation by an upstream kinase, and so if we eliminate that kinase, we may eliminate this particular ability of CK2 to phosphorylate PTEN. We conducted the screen using phosphorylation of PTEN as readout in HeLa cells. HeLa cells are not TLL cells, as you probably know, they're cervical cancer cells, but they were very convenient for a number of reasons. So we did the screen in HeLa cells, and we came up with this gene here called CHEK1 as a very interesting candidate for regulation of CK2-mediated PTEN phosphorylation, meaning that CK2 in HeLa cells, when we silenced it, when we knocked it down, we lost phosphorylation of PTEN at this particular residue that we know it's CK2 dependent. So this sort of, we were very excited with this, very happy, and then turned out and then tried to analyze TLL cells and see what was going there on there. The problem was that in TLL cells, I'm not showing you any of the data here, but because I want to show what happened next. In TLL cells, we didn't see the same thing that we saw in cervical cancer cells. So CHEK1 was not positively regulating CK2 in its ability to phosphorylate PTEN. Now, this could be the end of the story, but we are sort of stubborn people in the lab. So we looked into the literature and found evidence in a paper that was published in Nature Medicine a couple of years back. I think you guys probably are not seeing the references that I have here below, unfortunately. But so this paper that came out in Nature Medicine was indicated that oncogene activation, this particular oncogene called TLX, which is a transcription factor, would lead to downregulation of CHEK1, and downregulation of CHEK1 led to genomic instability and to transformation in TLL. So this kind of makes sense because CHEK1 is very important for something that we call, for the regulation of something that we call replication stress. Now, to put it very simply, at the molecular level, replication stress is when the replication fork sort of stalls, it has to stop and leads to the appearance of single-stranded DNA that has to be somehow dealt with. So if there were no signals to cope with this, the consequence would be precisely what I just told you, genomic instability, because after the single-stranded DNA that shows up, you probably are going to end up with DNA double-strand breaks, and these are hell. Okay, these are really good for the creation of genomic instability. Fortunately, there is this axis here between ATR and CHEK1 that serves as an alert signal to the cell that says, we need to cope with replication stress and deal with this. Now, replication stress can be the result of exogenous treatments such as topoisomerase 1 inhibitors or hydroxyurea, but it can occur also as a consequence of oncogene activation, particularly those oncogenes that trigger proliferation. And the reason is that, again, being a bit simplistic, you can imagine that if you have a very powerful oncogene pushing the cell towards a division, the cell may not be able to cope, to be as fast as the oncogenic stimulus, and so replication may sort of lag behind. And if that's the case, then you have replication stress. So all of this to say that CHEK1 really is important for prevention of genomic instability, and so it's viewed as a tumor suppressant. Now, again, despite all this, we still decided to go and look at CHEK1 levels in TLL and see if our results conform to what is in the literature. And to our surprise, really, what we found was something, again, quite puzzling, was that, in fact, in TLL primary patient samples or in TLL cell lines, as compared to normal thymocytes, again, the levels of the CHEK1 transcript, so this is the name of the gene, of CHEK1 transcript are actually higher in the leukemic cells as compared to normal thymocytes. And they're the highest in these TLL subgroups that are called TAL-LMO and the proliferative subgroup, that are those that are known to have higher proliferative indexes. So which really makes sense if we think of replication stress. Now, what we found at the protein level is that, again, comparing thymocytes with TLL cells, you know, thymocytes have very low levels of CHEK1, barely detectable here, very low here, whereas, you know, TLL patients and cell lines really have high levels of CHEK1. Not only high levels of CHEK1, but active CHEK1. So this is the phosphorylated residue that indicates activation. Also, activation of the upstream kinase that regulates CHEK1, ATR. You see it in the TLLs, you don't see it in the normal thymocytes. So really this indicates that, for some reason, contrary to what had been described before and to what one would expect, CHEK1 is actually upregulated, activated in TLL, human TLL samples. Mind you, this Nature Medicine paper was essentially conducted in an animal, very elegant, very cool, but in an animal model. Now, to try and understand whether functionally CHEK1 was really important or not for TLL cells, what we did was to use a pharmacological inhibitor of CHEK1 and see whether the cells were sensitive to CHEK1. And what we found was that leukemia cells are very sensitive. So this is the drug response curves, and they are very sensitive in the nanomolar range. You know, here in this particular cell lines, in the low nanomolar range, to inhibition of CHEK1. Mind you, in other cancer cell types, namely, you know, epithelial cancers, CHEK1 inhibitors were used in combination with topoisomerase inhibitors. And the cells were, you know, the IC50s were in the high micromolar range, or, you know, at least in the mid micromolar range. So it's quite different. These cells are very sensitive. And why is that? In fact, the evidence we have, what I showed you already and what I'm showing you here in this slide, is that this is due to high levels of replication stress. Why is that? Because if you look at what we call gamma H2AX foci, which are here in pink in these cells, upon treatment with the CHEK1 inhibitor, these foci, which indicate the existence of DNA double-strand breaks, really go up when we inhibit CHEK1. So what this indicates is that here, gamma 2AX foci are quite low, because CHEK1 is putting replication stress under check. When we release replication stress because we no longer have CHEK1, then we have a boost in gamma H2AX foci. And in accordance with this, what we found when we looked at the cell cycle distribution, so if you look here at these histograms, this basically this early peak, for those that are not too acquainted with this means that the cells are in G0 or G1 of the phase of the cell cycle normally, so they have 2M DNA content. Here, normally, this is cells that are replicating, so between 2N and 4N DNA contact, so they are supposed to be in S phase. And this peak here are cells that are starting to enter G2 or are already undergoing mitosis, so they have a 4N content, but they do not divide yet. Okay, so what we found, which was really quite amazing, I would say, is that when we treat the cells with the CHEK1 inhibitor, the amount of cells that are with an intermediate DNA content that we would normally say they are in S phase really increased no matter what the cell line. You know, if you compare this with this, or this with this, or this part here with this, really indicates that you have a lot of cells that are replicating but did not finish replicating. Okay, and you could say, okay, they did not finish replicating because they are still, you know, in S phase, so that's cool. But the evidence we have is that they are actually, or at least many of them are actually already entering G2M and mitosis. Why? Because there is an increase upon the use of the CHEK1 inhibitor of CDC25C and a decrease in phospho-CDK1, this residue. And the combination of these two findings really indicates that the cells are entering. These are sort of hallmarks of cells that are entering mitosis. So what we have here, it's evidence that the cells are entering mitosis with an aberrant DNA content that is not 4N. Okay, and normally what this leads to is what we call mitotic catastrophe. The cells cannot cope with this, and so they will enter apoptosis. And that's what we see here. So maybe you can focus just on this quadrant here, which are the live cells, and you can see that cells, you know, in this cell line, for instance, the viability drops from 89 to 49%, and you see always the same trend. So there is a real push towards cell death and towards apoptosis. And if instead of using a pharmacological inhibitor, we go and silence CHEK1, it's again the same thing in terms of cell cycle distribution. And now, mind you, this is the scramble control and this is the knockdown. Let's focus, for instance, at 96 hours of treatment. 60% viability with the scrambled control, with the knockdown with the shRNA against CHEK1, you can see that there is very little cells alive. So the treatment, pharmacological treatment or genetic elimination of CHEK1 really do the job in terms of promoting cell death of TRL cells. Now, is this tumor specific or will we, you know, kill any cell, and particularly the normal thymocytes? Well, what we found was that in fact, because normal thymocytes are not undergoing these massive levels of replication stress, if you look here at the black circles, these are normal thymocytes. This is the viability index. Okay, the highest, the more viable the cells are, and you can see here that normal thymocytes are, you know, they may be sensitive at high doses, but not that sensitive. Whereas, you know, at lower doses, in particular, in intermediate doses, you can see that there is already an effect in TRL patient samples and not in the normal thymocytes. And in any case, at higher doses, the difference is huge, as you can see. So this really sort of made us very happy, and we thought this may be a very interesting drug to explore in the clinic. So to try and push in that direction, again, we went for an animal model. At the time, I didn't have any major grants that financed the lab tremendously, so we had to be very strict on the kind of experiments that we did. And so we did this sort of experiment in which we treated the mice for only a period of time. But as you can see here, again, the use of the CHEK1 inhibitor can delay leukemia progression quite significantly. And so I would very much like that we would explore, that pharmaceutical companies would explore targeting this axis in TLL. I'll get back to this in a few minutes. But let me summarize what we found. So basically, we found that in TLL, oncogene activation leads to high levels of replication stress that are put at bay, under control, due to this axis here, ATR check1. And this is what allows replication stress to be under control, and because of that, there's no significant apoptosis. If we inhibit check1 or we silence check1, then replication stress is exposed, the cells cannot cope with it, and they undergo apoptosis. And this is leukemia specific, it doesn't happen in normal pharmacytes. So if we put things in a more integrated view, yeah, perhaps this paper that I told you about was right in that, at least downstream from some oncogenes, check1 needs initially to be downregulated. But after a while, or depending on the oncogene that is upstream, check1 really needs to be upregulated in order for the cells to cope with replication stress and to be viable and proliferate. And mind you, at this stage, coping with replication stress also associates with decreased genomic instability. Now, Ramiro, I'm sorry, can you tell me how much time I have? Because I really think I need to rush.
None, it's all gone.
Then I will, if it's all gone, I will skip. I just wanted, am I allowed to have 2 minutes?
Of course. Please just conclude as briefly as you can.
Yeah, so what, I think what I've just told you illustrates something that really concerned us, which is the fact that we had a lot of lab work done trying to explore the mechanisms by which TLL occurs, trying to identify molecular targets for therapeutic intervention. But because we did not generate the tools to target the leukemic cells ourselves, we were we were highly dependent on pharmaceutical companies or the programs of other universities and so on to define what's the clinical program in terms of translation of results into clinical applications. And so from a point on, what we decided to do was really to try and develop our own tools that we can, we become more entrepreneurial in a way. And so what I want to wrap up with is just 2 things. Actually, it's cool because for those that were in my previous talk, they know this already. But because this axis between IL-7 and IL-7 receptor is very important for leukemia progression, and let me just focus on this slide here, at least 70 to 80% of TLL patients may benefit from therapeutics that target this IL-7, IL-7 receptor axis. We developed antibodies, and we are developing currently antibodies to try and target leukemia samples with our own proprietary stuff, okay, that we can take to the clinic if we are able to. The other thing that we are trying to do, and I have to share this with you, although I can't show much, but because I think it's quite cool as an idea, we're trying to explore the fact that, microRNAs, which are these around 22 nucleotide long small non-coding RNAs that every cell has, but they are differentially expressed in cancer cells versus normal cells. So and they are sort of expressed in a tissue-specific manner. So we are trying to devise what we call a microRNA detector system, which will identify the leukemia or cancer cells in general by their microRNA or by a particular microRNA expression profile that will then regulate the expression of a therapeutic gene that will selectively kill the cancer cells without touching the normal cells. So this is the rationale. I can't say for IP reasons how we are doing this, the results are very, very promising. We're very excited with this, and I hope to share with you guys later on, sometime in the future, if you want to invite me again for something else. In any case, this is it. I would just like.
João, I think we lost him. Definitely.
That did the work.
João.
Sorry?
We didn't hear you for like 10 seconds, so if you don't mind, say it again.
Sorry. Yeah, so I would just like to thank you all for your attention, and thank the people that actually did the job, thank the collaborators without whom none of this was possible, thank the funding. Without funding, science is not possible, and I hope the European Union realizes that at this very critical period in our life. And with that, I finish and take any questions if you have.
Thank you very much, João. It was very, very interesting. I'm sorry I didn't warn you that time was running out. I was so enthralled in your explanation that I forgot that. So is there any very, very, very brief question that anyone want to pose, João? Or I would defer to a query outside this forum. I'm pretty sure that João, if you send him an email, he'll answer back ASAP. So let me say and finish this section of the workshop by thanking both the prelectors. Thank you, João. Thank you, Iola. It was very, very interesting. I would like to also thank the organization for inviting me to be here today. And I hand over the session to Ana Sousa, if I'm not mistaken.
I think by now Mara would like to finish the session, and maybe we can ask all of the people from the organizing committee. But first, I really want to thank Iola and João for accepting the invitation and for being here. We know that it's not easy to do online presentation. It's so much better to do it on person. And I'm sorry for not having time for the questions, João. Next time we will for sure discuss more. Mara?
Hello again. Well, thank you so much for sharing this session and for the two speakers of this slot, the last slot of the afternoon. And I would like to ask first to the organizing committee to turn on their cameras. They deserve it. All of them, they work a lot. So, well, we reached the end of our workshop, and the words that I will say, I think they express our feelings from all the organizing committee. Well, we are quite happy, and we shared some message during the day, and we are quite happy with the final result of the workshop and hope that it will be valuable for most of you. We think that we covered a series of different topics in the cancer framework. Some were difficult for some of us, so it's different areas from our expertise. But I do believe, and we do believe that our collaborations, I mean the collaborations between our research group and the groups from the invited speakers will be reinforced. But also we do believe that new ones will be created between the different researchers attending and presenting in this workshop. And we saw several examples of discussion between the invited speakers, and we are quite happy for that. And before ending, I would like to deeply acknowledge our invited speakers for the relevant talks provided, namely, and I have here the list, from the beginning and by order of presentation, they were João Conde, Joana Rodrigues, Mateus Pereira, Teresa Gouveia, Francisca Silva, Cátia Venâncio, Susana Viegas, Luís Branco, José Esperança, Iola Duarte e João T Barata. So thank you so much. We also would like to acknowledge to our chairs. They were João Rocha, Isabel Lopes, Pedro Carvalho and Ramiro Pastorinho. We acknowledge also all participants that were with us up to the end. So we are recording for 9 hours. This has been done for 9 hours. It's more than a day work, normal day work. And also finally acknowledge, and this is in my own, I would like to acknowledge the organizing committee. They were unstoppable, and they are Ana Souza, Márcia Neves, Francisca Silva, Mariam Colani, and Rita Teles. So thank you all. Have a nice evening and a wonderful week ahead. Thank you so much. And see you. I don't know if someone wants to add something or if we can consider that the workshop is ended.
You should go now for the wine tasting that we had in the...
Well, we will do it at home.
next year.
See you. Bye-bye.
Bye-bye.
See you. Bye.
Bye. Bye. Bye-bye. Nice to see you all.
Bye.